Hello everyone, welcome to the A9 Study Center. Today I want to share some knowledge regarding the fish reproduction, induced breeding by hypophysation and hormones. The traditional methods of induced spawning for crabs are based on injection of crude extract of common crabs pituitary glands, that is hypophysation. Major crabs and other culturable species of fish generally breed in running water in rivers during monsoon with several environmental favorable conditions for spawning. These fishes become sexually mature in ponds but do not spawn. The rat breeders can be induced by spawn by administration of pituitary hormones, which stimulates the fish to release eggs and sperms. This technique is based on the fact that the gonadotropic hormones, that is FSH, follicular stimulating hormone, and LH, luteinizing hormone, secreted by the pituitary gland, that help in maturation and spawning of fishes. So this technique is called hypophysation. The Brazilians were the first to develop this technique in 1930, and followed by the Russians, who successfully applied this technique to breed sturgeons. In India, Khan 1938 successfully induced Cineus myrgala to spawn by injecting mammalian pituitary hormone. Other species that have been bred by hormone injection are Heteroneuts fossilis, Clarius bratacus, Leviorohita, etc. The advantages of this technique are as follows. A. Pure spawn and fry of a desirable species is available for culture. This method ensures timely availability of fish meat in required quantity. It is economical as it eliminates the cost of holding potential spawners for a long time and the expenditure incurred on collecting fish meat from rivers is much more than on induced spawning by hormones. Number D. The method is simple and can be easily practiced even by a layman. Procedure of induced breeding. First, collection of gland. In fish, the pituitary glands lies in a well-protected depression myodome on the floor of the skull. For success in induced spawning, pituitaries should be obtained from the fully mature healthy donor fish. Generally, pituitary from the fish of the same species or from a phylogenetically related one is preferred, but hormones from an unrelated fish can also be used. Glands from either male or female fish can be used and are equally effective, but little qualitative difference between the sexes. The pituitary gland can be collected by any one of the following means: A. Through the foramen of magnum. B. By dissecting and cutting the roof of the skull. A. Through the foramen of magnum. Collection of gland through the foramen of magnum is easier and economical as the fish head can be sold after taking out the pituitary. Next point. The foramen of magnum is first exposed by removing muscles and the parts of vertebrae left with the cut head. Next, the fatty substance covering the brain is removed by means of a forceps and then by means of a piece of cotton, taking care not to damage the brain. Next point, the anterior part of the brain is now detached by means of a fine forceps, and then the brain is carefully taken out of the foramen. Next point, the pituitary gland is exposed, clean the membrane and picked by means of tweezer and kept in a petri dish. B. By dissecting and cutting the roof of the skull. For collecting the gland by dissecting the head, the roof of the brain cover is cut by means of a sharp butcher's knife. Next point, the dorsal side of the brain is now exposed by removing the fatty substances with a piece of cotton. Next, the olfactory and optic nerves are now cut to free the brain and carefully lifted off and placed in a petri dish. Next, the pituitary situated behind the optic chiasma is exposed and removed. Point number 2. Preservation and storage of gland. Pituitary gland can be used immediately after collection in fresh condition, but is usually preserved and stored for future use. The gland, if properly preserved, is known to retain its potency for several months. It can be preserved by any one of the following methods. By freezing, fresh gland is immediately frozen after collection and kept it in a refrigerator in frozen conditions. In absolute alcohol, fresh gland is kept in pure absolute alcohol in a phial. After 24 hours, the alcohol is changed and the phials are kept at room temperature or in a refrigerator. The phials must be airtight to prevent moisture from getting in, or the phials are kept in a desiccator containing some anhydrous calcium chloride. Absolute alcohol defattens and dehydrates the glands and should be changed occasionally to maintain the gland in good condition for a long time. Preservation in acetone. The gland is put in fresh acetone or ice-sealed acetone in a phial immediately after its collection. It is kept in a refrigerator at 10 degrees Celsius for 36 hours, during which acetone is changed after every 12 hours. The acetone defattens and dehydrates the gland, which is then taken out on a filter paper and dried for 1 hour at room temperature. The dried gland is then accurately weighed and stored in a phial in a refrigerator for future use. Preparation of extract and its preservation. For giving injection to the breeders, previously wet glands are taken out and homogenized in distilled water or 0.3% saline water. The suspension is centrifuged and the supernatant fluid is used for injection. However, it is better to prepare the extract in advance and preserve it for injection when needed. For this, the extract is preserved in pure glycerin in airtight phials. The ratios of distilled water and glycerin should be 1:2 and a concentration of 20 to 40 mg of gland in 1 ml of water, and the glycerin is considered to be most convenient. After 3 to 4 days, sediments settle down in the bottom and the solution is filtered, and the filtrate is stored in a sealed glass ampoules. Selection of breeders. For success in induced breeding, it is very important to select good, healthy breeders well in advance and kept them in breeding ponds. The breeders should be of medium size, that is 1 to 5 kg in weight, and in the age group of 2 to 4 years. Next point, the breeders are given balanced diet for healthy growth of the body and attaining advanced gonadal maturity. Next point, they are fed with cheap artificial food consisting of rice bran, oil, cake, powdered cotton seed, etc. daily for 15 to 30 days before injection of the pituitary extract. And then before the commencement of the rainy season, males and females are segregated in separate ponds. Next, preparation of injection and doses. The breeders are selected carefully and weighed by means of a spring balance so that the quantity of the gland to be injected can be determined. One set of breeders usually consists of 1 female and 2 males. Injection of the predetermined dose is given with the help of a hypodermic syringe, either intramuscularly or intraperitoneal. Intramuscular injection is easier and usually administered on the caudal peduncle or near the shoulder region. Intraperitoneal injection is given at the base of the pelvic fin or sometimes at the base of the pectoral fin. Care should be taken not to damage the internal organs of the fish. The dose of the pituitary gland is calculated in relation to the weight of the breeders to be injected. This is important for the success of the experiment, but is a difficult matter as the potency of the gland is influenced by the size, stage of maturity, and age and sex of the donor fish. The dose also depends on the size and stage of maturity of the recipient fish. Usually, a preliminary dose of 2 to 3 mg of pituitary gland per kg body weight of the fish is given to the female fish. Males do not need any preliminary dose as they push milk on slight pressure. Next point, after an interval of 5 to 6 hours, a second dose of 5 to 8 mg of pituitary gland per kg body weight is given to the female, while the males are given a dose of 2 to 3 mg per kg body weight. Or a single high dose of injection is also given at the rate of 7 to 12 mg per kg body weight of female and 2 to 3 mg per kg body weight of the male breeders. Assessment of proper dose for induced breeding of fish is very important, as low doses may not be effective, while overdose inhibits spawning or causes premature release of ova which do not fertilize. After the injection, the breeders are released immediately in the breeding hapa or cistern. Use of natural and synthetic hormones. Induced hormone of crabs and other species by hypophysation is a popular and reliable method for obtaining pure seed of desirable fish species. However, it is difficult to collect large quantity of the gland from fully mature fish for commercial use. So the pituitary extract has to be administered into separate doses to female fish for achieving successful spawning. This is not only resulted in increased handling of brood fish, but often leads to spawning failure, mortality, and a loss of time and labor. Hence, the need for safe ovulating agents to replace the pituitary extract was felt, and some purified natural and synthetic hormones have given positive results. Of this, mammalian chorionic gonadotropin (CG) has given good results. Spawning has been induced in Cyprus carpio, Sirenius myrgala, carriers, Bradagers, etc. by administering human chorionic gonadotropin (ICG), purified ICG, proline gonadotropin, and human pregnancy urine have also been successfully used in bringing about spawning in several species of fishes. Good results have been obtained by using a mixture of CG and mammalian pituitary extract known as cynorrhea in combination with fish pituitary extract. Spawning could be induced in H. fossilis by administering a high dose of pregnant mare serum (PMS). Several species of crabs and catfish could be bred by administering the drug like obiprem or obitate at room temperature. It contains gonadotropin releasing hormone in solution and can also be used in combination with pituitary extract for better results. Besides these hormones, certain gonadal steroids such as the deoxycorticosterone acetate (DOCA), 17-deoxycorticosterone, and corticosterone can be used as a substitute for pituitary extract for inducing spawning in fishes. Thank you for watching.